and welcome to the second MDBI webinar on COVID-19. Today, we are happy and privileged to have a legend on this topic. Professor Susan Weiss. Susan Weiss is an American microbiologist who is professor at microbiology at the Perelman School of Medicine at the University of Pennsylvania. Her research considers the biology of coronaviruses, including SARS, MERS, SARS-CoV-2, and as of March 2020, she serves as co-director of the University of Pennsylvania Coronavirus Research Center. During the presentation, you can post your questions at the Q&A window, and if you want, you may also raise your hand to signal that you want to go live. If you are a researcher and interested to submit a paper in one of our supporting journals, IgE, RPH, and viruses, please get in touch with us. A list of open special issues will be sent to you after the webinar. And now I would like to pass the word to Susan. Please enjoy.
Okay, thank you so much for having me. And I'm hardly a legend. Okay, so I'm going to give a very introduction lecture to coronaviruses, kind of divided into three parts. I'll give a brief history of coronaviruses with a focus on the human viruses, then talk about the biology of the viruses, of the replication of the life cycle, and then talk a little bit about the ability of coronaviruses to antagonize innate immunity, and that'll include some work from my own lab. The rest of it will be kind of history and background information. So these are coronaviruses. They're members of the nidovirus superfamily. And nidoviruses are named for the nested genomic mRNAs generated during infection. Nido comes from the word means nest. These are all envelope viruses. They have membranes. They're kind of medium-sized, 100 to 150 nanometers, and they have single-stranded positive sense RNA genomes. And these are, other than coronavirus, this group includes arterioviruses, roniviruses, and toroviruses. And here's a picture of them. And the coronavirus gets its name from its corona-like spikes, the corona-like morphology, and that's due to these spike proteins in the membrane. I think everybody's heard about the spikes, and we'll talk about them some more. So this is a diagram of the coronavirus particle. It's a very simple particle, really. It has a very long RNA genome, a non-segmented genome of 30 kilobases, the longest RNA genome known. It has a cap 5' end and a polyadenylated 3' end, and as such serves as a messenger RNA. Surrounded, and the RNA is complex with a nucleocapsid protein, forming this helical structure, or helical nucleocapsid inside of the virus particle. Around the nucleocapsid is a membrane, shown here, that's derived from the host cell when the virus buds out of the cell upon assembly. And within that membrane, there are all coronaviruses have three proteins. The spike protein, which everyone's heard of, I think, because the spike protein is the protein that mediates entry. It binds to the receptor and mediates fusion of the viral membrane and the cell membrane. It's also a major determinant of tropism of the T cell and B cell responses and of virulence. But also in the membrane are two other proteins called membrane protein and small membrane protein, and they also have important roles in the virus life cycle. And in addition, some coronaviruses express a hemagglutinin esterase protein, which is kind of like the flu HA, but SARS-CoV-2 and its relatives don't express this virus. So it's a non-essential protein for coronaviruses. So before 2002, that's when the first SARS emerged from in China. Most coronavirus research focused on murine coronavirus, which was isolated in 1949. And this virus has been studied for really quite a lot of time in a lot of depth because it presents us with a really nice model for hepatitis, encephalitis, and chronic demyelinating diseases, has been used as a model to study the human disease multiple sclerosis. And a lot of what we know about coronaviruses, a lot of the mechanisms of replication, about entry, about pathogenesis, really come from these early studies on murine coronavirus, MHV. In addition, animal coronaviruses were studied for quite a long time. They're important pathogens of pigs, cows, cats, birds, and there's a lot of vaccine development that was done with these viruses way back. I found references to to IBV, the chicken coronavirus, back into the 30s. And so these two were two of the main foci of studies of coronaviruses until SARS was emerged. We also knew there were human coronaviruses, so the types of diseases known to be caused by coronaviruses, again, before SARS, there were respiratory viruses, a human OC43 and 229E. There was not a lot of research on these viruses, but they were clearly identified as human cold viruses. The avian IBV and the bovine BCV also cause respiratory diseases. The mouse viruses, some of the MHV strains cause central nervous system disease, as I said before. Some MHV strains cause hepatitis, liver infection. Many of these viruses are gastrointestinal viruses, the porcine transmissible gastroenteritis virus, the avian IBV, the bovine BCV, MHV as well. The mouse strain, the strains that are found in mouse colonies are GI viruses. And then there's the feline coronavirus. And also there's another feline coronavirus called FIPV, feline infectious peritonitis virus, this is a really fatal, very severe disease of cats. So there really was a whole literature and a whole lot of coronaviruses that were known and were studied far before we knew about the lethal human coronaviruses. So this is a timeline of coronavirus kind of history starting in the 60s and 70s, although as I said before, we did know about some of these viruses before that. So I would say during this period from the 60s until the present, people have been studying the MHV model, the animal viruses, the vaccines, and human cold viruses. And that's continued even after the emergence of the lethal viruses. So just to summarize again, OC43 and 229E are two common cold viruses. They infect the upper respiratory tract, but OC43 can also occasionally infect the lower respiratory tract and cause more severe disease. The next human coronavirus that we knew about was SARS coronavirus, which emerged in China in late 2002. And then around that time, people started looking for other human coronaviruses. I call this the era of pathogenic human coronaviruses. And HKU1 and NL63 were identified as human coronaviruses, HKU1 causing pneumonia and NL63 causing croup and bronchiolitis. So these viruses cause more severe diseases than the cold viruses, but certainly much less severe than SARS. Around the same time, this is when it was discovered that there were many coronaviruses found in bats, and that in fact SARS was probably a descendant of a bat coronavirus. And then it was pretty quiet in coronavirus research fields until about 2012, when MERS, Middle East respiratory syndrome virus, emerged in the Middle East. And causing a similar disease to SARS. And then again, things kind of quieted down. And then at the end of 2019 and now in 2020, we have this pandemic with SARS-CoV-2. So just to be clear, SARS-CoV, the original one, MERS-CoV, and SARS-CoV-2 all cause severe respiratory diseases. And COVID19 is actually the name of the disease caused by SARS-CoV-2, coronavirus disease 2019. So just to distinguish between, this is the name of the virus, SARS-CoV-2, and COVID-19 is the name of the disease that it causes. Just a little more history, the first International Coronavirus Symposium was assembled in Würzburg, Germany in 1980, and there were about 60 people at that meeting, and that was pretty much the whole field. And then in 2003, the 10th International Coronavirus Symposium was convened in Egmond am Zee in the Netherlands. And that had a lot more people in it. This was just right after SARS had emerged. So this meeting attracted a huge number of people. And a lot of people came into the field of coronaviruses. And then this year, there was this 15th International Coronavirus meeting, now called Nidovirus meeting, was to be held in the Netherlands in the same place as the 2003 meeting, only this meeting was postponed because of SARS-CoV-2. So it's just kind of an eerie coincidence to me that each of these meetings was going to be held in the same place, just at the same time of the emergence of one of the SARS viruses. So hopefully this meeting will be held next year. That's the plan. Okay, so I want to talk a little bit about SARS interspecies transmission or coronavirus interspecies transmission. So I think by now it's well accepted that SARS coronavirus originated in a bat and was transmitted to a civet and then transmitted to humans. And then once in humans, it spread very quickly by human to human close contact. And this is a map of where SARS infections occurred. The vast majority were in China and Hong Kong, and they did spread somewhat, but I don't think there were any deaths from SARS in the US. There was a kind of epicenter of SARS in Toronto, as well as in Asia. And this whole epidemic was over in 8 months. There were a little over 8,000 infections with close to 10% mortality. And as I said, about 87% was in China and Hong Kong. So this was a really very severe disease with higher mortality than SARS-CoV-2, but it was really restricted and ended really quite quickly, only maybe about 8 months. And we really don't know how many times this transfer happened or how long this virus was around in civets before it went to humans. Did this only happen once? We don't really know. And it's kind of surprising that we didn't see more evidence of more SARS, but it's really been gone as far as we can tell since 2003. So MERS had a little bit different pattern. It also is pretty clearly comes from a bat, a different kind of bat from the SARS viruses. It has a reservoir in camels. It's pretty clear now that camels in both the Middle East and in Africa, many of them have evidence of MERS infections. The camels can transmit to other camels and also to humans. A lot of times people that are working with camels, camel handlers. And then there is human to human spread of MERS, but not nearly as much as there was with SARS. And most of MERS, I think almost all of MERS is in the Arabian Peninsula, except for an epidemic that occurred in Korea by somebody moving or visiting from, returning home from the Middle East to Korea. So different from SARS, camels are actually a reservoir for MERS, whereas we think the civet was just kind of an intermediate species and maybe doesn't really harbor lots of SARS. Also different from SARS, there are still new cases in 2019. Anyway, I haven't looked at 2020. There are a total of 2,400 cases, and this one is even more lethal than SARS or SARS-2, causing about 35% mortality. So this is sort of similar to SARS, but really different as well. And SARS-CoV-2 is actually behaving differently from either of the other viruses. We believe it has a parent in the bat, although that has not been identified. We think there's an intermediate species, and it's been rumored to be or suggested to be a Malayan pangolin, but this really hasn't been proven at all. And I don't think there's any really hard evidence that that is the species. And then it clearly was transferred to humans in Wuhan, where it caused a major epidemic. Very fast human to human spread, very contagious. And this time it really did spread all over the world. Here's just a map, as we all know, because we're all living with this virus. This is a month old. I should change this. So there were over 400,000 infections and 19,000 deaths from SARS-CoV-2. And that's the reason, obviously. And the US alone had 140,000. I'm sure this is much higher now. I should really check out those numbers. But so this is the reason why we're giving online seminars and not in-person seminars. So just to put it into perspective, the coronavirus cross-species infection was really known about before SARS. For example, the human coronavirus, OC43, and the bovine coronavirus, BCV, are very, very closely related. They're also closely related to the mouse virus. So it's believed that the human OC43 may have evolved from the mouse virus adapting to humans, shown here. And it's also clear that the bovine virus moved into alpaca and ruminants, where it adapted to those species. And we now have alpaca and ruminant coronaviruses related to the bovine virus and the human virus. So the cross species is not something particular to SARS and MERS, but maybe a more general coronavirus characteristic. So where did SARS coronavirus 2 come from? I'm going to talk about some of these things. What genera or lineage of coronavirus? What is its parent virus, and what is the intermediate species? And of course, we don't really know the answers to these two, but I'll talk about some of the data that's been used to try to figure that out. So this is pictures or diagrams of coronavirus genomes of the lineages that contain the human viruses. And that includes alpha coronaviruses, of which 2290 and NL63 are members, and three sublineages of beta coronaviruses. Lineage A, which includes OC43 and HKU1. Lineage B, which includes SARS and SARS-like bat viruses, and lineage C, which includes MERS and MERS-like bat viruses. And all these genomes are arranged very similarly with a very long replicase locus that encodes 16 non-structural proteins. And I'll talk about that in more detail in a little bit. And then downstream, a spike E, M, and N, always in the same order with all the lineages. So the gene structure or organization is quite similar for all the coronaviruses. The difference is these small proteins here with numbers, 4A, 4B, 5A, et cetera. And these are the so-called accessory proteins. They're not required for replication in tissue culture, but they often have host antagonist activities. And I will talk about that more later too. And that's one of the things that really distinguishes these lineages, the arrangement of the non-structural proteins. And it looks like the ones for SARS have the most complex. So of course, pretty soon after SARS-CoV-2 arose in China, in Wuhan this year or last year, it was sequenced, and it was clear that it's very closely related to SARS-CoV, the first original SARS-CoV, and that's why it was named SARS-CoV-2 for its homologies to SARS-CoV-1. There are some differences. Places to look for differences are the spike, which I'll talk a little bit more about later too, and also the accessory proteins, which are quite different from those of the other viruses. So just two things about coronaviruses that have something that may be interesting in terms of the diversity or where they come from. Coronaviruses undergo discontinuous transcription, and I'll talk about that more in detail later too, leading to a high recombination rate. So new viruses can arise by recombination. But on the other hand, the error rate for RNA replication is reduced by a proofreading enzyme called XON or NSP14, and I'll talk about that later too. So we really don't know the origin of SARS-CoV-2, but we can think about maybe there's a recombination event that led to this virus, but we don't know that. So this is from a paper from Zhengli Shi's lab very early on in the coronavirus epidemic. And what she did was compared it to a bunch of other viruses. And the virus, so this blue line here shows very close homology to SARS-CoV-2. And that is a bat coronavirus called RATG13. And I think so far that virus is the closest one in homology to SARS-CoV-2, although there are some important differences in the spike protein. And here is, so SARS itself, human SARS, is not very closely related. It's one of these lines here. And then this is another bat virus in pink. So there are a couple of bat viruses to which SARS-CoV-2 is closely related, more so than it is to SARS. And yeah, it's about 96% homologous to this bat CoV RATG13. And here's a homology tree based on open reading frame 1B, the replicase locus. And we can see here that SARS-CoV-2, these are all beta coronavirus from Wuhan. This is now called SARS-CoV-2. They're very closely related to this bat virus. Whereas SARS-CoV-2, original SARS is up here. So it's closely related, but much further away than the bat virus. And then here's MERS coronavirus, and then OC43, and then 2290, the alpha coronavirus. So this is clearly in this group of beta coronaviruses. Oh, NL63 up there too, sorry. Okay, so let's talk a little bit about the spike protein and the receptor utilization. So SARS coronavirus receptor is ACE2, and interestingly, NL63, the croup virus, also uses ACE2. This is an alpha coronavirus, beta coronavirus, not very closely related, but they do use the same receptor, but cause very different diseases, which always reminds me that the receptor utilization is not really the whole story about what makes something virulence or what what is the only determinant of tropism. MERS coronavirus, on the other hand, uses a completely different receptor, DPP4, dipeptidyl peptidase, and causes a kind of similar disease. So again, receptor is not the entire story. SARS-CoV-2 also uses ACE2 receptor. The spikes of these two SARS are 75% identical, but importantly, they differ in some key residues of the receptor binding domain, the part of the spike that actually binds to its receptor. And I guess it's been speculated that the virus is still adapting to humans, and I don't see any evidence that it's changing very much with all the sequencing that's been done, but we will see if that happens. And there are also data suggesting the spike of ACE2 of SARS-CoV-2 binds more strongly to ACE2 than the spike of SARS-CoV-1. Okay, so let me just talk a little bit about the spike protein. So here's a diagram of this coronavirus spike protein. They're all quite similar. There's a receptor binding domain. I should say it's divided into two subunits, S1 and S2, amino terminal, carboxy terminal. S1 contains a receptor binding domain. For some viruses, it's at the extreme end of the protein, but for SARS, it's over here. There's a fusion peptide here that mediates cell-to-cell fusion. So in order for this spike protein to be active to mediate fusion between the virus and the host, it has to be cleaved by proteases in two places, at the S1, S2 border here, and at the S2 prime border here, right next to the fusion peptide. And just in comparing all these coronaviruses, the lineage B and lineage C, A, B, C, and the alpha coronaviruses, the human coronaviruses, we can see here that some coronaviruses like OC43 and MERS have this so-called furin site here, right here, which would be at the S1, S2 border. And that's, whereas that's shown here, whereas the lineage B viruses such as SARS and the SARS-like bat viruses are missing this site. And the significance of this is that viruses with a furin cleavage site here may enter cells by a different pathway than viruses that don't have it. And I'll talk about that a little bit later again. But importantly for here is that that this furin site may help us identify where SARS-CoV-2 came from, because as I said, here's the furin site. It's missing in both SARS itself. It's also missing in the bat virus that we just talked about, ATG 12. So let's look a little bit more at the spike protein. So here's the spike protein of SARS-CoV-2, and here's the S1, S2 furin cleavage site, and the second S2 cleavage site here, the fusion peptide. So this is a simpler way of looking at the protein. And here is the protein from the pangolin. So as I said before, there's been a lot of speculation that there may be a pangolin coronavirus that's a precursor to SARS-CoV-2. And indeed, the receptor binding domain of the pangolin virus is very closely related to that of SARS-CoV-2. But the entire spike protein is not. It's about 90% related. And again, and the furin site is not present, this telltale furin site in SARS-CoV-2. And in RATG13, the receptor binding domain is quite different from that of SARS-CoV-2 in the pangolin, but the whole spike protein is about 96% related. And again, there's no furin site. So what this data says is that we really don't know, we don't have a telltale spike protein in another coronavirus that might tell us where SARS-CoV-2 comes from. So I do think that finding a virus with this furin site in a bat would be helpful in trying to track down the origin of SARS-CoV-2. So there are many questions remaining, and what is the bat ancestral virus for SARS-CoV-2, and what is the intermediate species? And was the origin, this is another question that I think's been speculated a lot now, was the origin of the outbreak in the Wuhan seafood market or elsewhere? What factors drive spillover into humans? Is it just proximity to animals, to bats or other intermediate animals? And why did it take 8 years for MERS and 17 years for SARS-CoV-2 to happen again? Or really did it? Were there really other spillovers into humans that may not have caused such pathogenic diseases so that we really didn't notice them? I think that could be quite likely. And this could happen again. So we really need to be thinking about going forward about what to do about it or how to prepare for it. Okay, so now I'm going to talk about coronavirus replication and try to point out potential targets for antiviral therapies. So this is a diagram of coronavirus life cycle that we published in 2005 after the SARS epidemic. And it's a very simplified diagram. We know a lot more now, but I think for just describing the life cycle, this is really kind of perfect. It's so simple. So the coronavirus particle, and this is for any coronavirus, attaches to its receptor. And then it's internalized into the cell by a virus cell fusion event, which I'll talk about a little bit more later. The nucleocapsid is released into the cytoplasm, and the RNA is copied into a polymerase, shown here. And that polymerase, it's copied into two long polypeptides called PP1 and PP1A1B. And from that, there's processed a replicase protein that's shown here with some other proteins that are important for replication as well. And that then copies more genome RNA and also synthesizes messenger RNAs. And I will talk about that in more detail later as well. But once these RNAs are made, they encode various proteins. This is actually the MHV proteins. And the glycoproteins, or the membrane proteins, M, E, and spike, assemble on intracellular membranes shown here. They identify viral regions of viral modification. Meanwhile, the RNA and the nucleocapsid protein complex to form nucleocapsids which bud into these membranes. They bud into a compartment between the endoplasmic reticulum and the Golgi called the ERGIC. So then these particles are found in vesicles, which are transported to the cell membrane and then released by a kind of reverse fusion event. So we get more virus here. Meanwhile, spike proteins are deposited along the cell membrane as well, and they can mediate cell-to-cell fusion, which is kind of a trademark or a hallmark of coronavirus infection. So I'm going to talk about two steps that have been identified as possible sites for antiviral interventions, and that would be entry and then the translation and the many replicase proteins encoded in PP1A and PP1A1B. So coronaviruses use two routes of entry into the cell, and a lot of the words here are probably familiar to you, but maybe the two routes of entry are not so familiar. So by the first route, which is called the endosomal route, the spike protein attaches to its receptor, which in the case of SARS-CoV-2, again, is ACE2, angiotensin converting enzyme. It then endocytosed into the cell, into an endosome shown here. And the next thing that happens is the spike protein mediates a fusion event so that the viral membrane fuses with this endosomal membrane and the nucleocapsid and RNA is released into the cytoplasm to begin the infection. Now, in order for that to happen, the spike protein has to be cleaved at both, as I told you before, at that S1, S2 border and at the S2 prime site. And in the case of this endosomal pathway, that cleavage is carried out by cathepsin protease, which requires a low pH environment in the endosome to carry out the cleavage. So, and then the second pathway is a direct plasma membrane route. And in this route, again, the virus spike attaches to the receptor. And in this route, it's cleaved by a protein by a membrane-bound protein called TMPRSS2. You've probably heard about TMPRSS2. And then that mediates the fusion of the spike, then mediates fusion through this plasma membrane. And again, the nucleocapsid is released into the cytoplasm for infection. Now, a recent paper I saw says that before this happens, the spike protein is actually cleaved by an intracellular protease by furin. So this route actually requires or uses the furin cleavage route, and probably requires that fusion, the furin site that I talked about that's present in SARS-CoV-2. So why is this important? This is important because coronaviruses can use different routes. They can use both routes. And this all depends on the protease sites available in the spike protein, and also the proteases that are present on the infecting cell. And that can depend on the cell type quite a lot. And these can be membrane-associated proteases like TMPRSS2. They can be in the extracellular space, or they can be the intracellular proteases like furin. And so, and why is all that important? Because when in designing antiviral therapies, you really have to think about what route you're targeting. For example, chloroquine that everyone's been talking about would only target this route because chloroquine raises the pH of endosomes and would prevent this cleavage, the cathepsin cleavage. And likewise, cathepsin inhibitors, for example, E64, would also inhibit this pathway. For this pathway, a TMPRSS inhibitor might work, the camostat mesylate, or a furin inhibitor. So because some viruses target both pathways, it might be important under some circumstances to really target both of these pathways. Although TMPRSS, there's a lot of literature now suggesting that TMPRSS is really required for entry in airway cells for SARS-CoV-2. Okay, I want to talk now about transcription of coronavirus RNA. So here's the RNA genome. As I said, it's a positive strand genome. It has a cap 5' end, a polyadenylated 3' end. It has a leader sequence, about 100 nucleotides, which I'll show you why that's mentioned. And then these yellow lines are transcription regulatory sequences, and they signal the beginning of each messenger RNA sequence and each sort of open reading frame for protein expression. So during infection, as I said before, an RDRP or RNA-dependent RNA polymerase is synthesized from the plus strand, from the positive strand genome. That just hops on the 3' end and synthesizes a negative sense antigenome RNA, shown here, and then gets on the 3' end of that RNA and synthesizes more genome. So this is how genome RNA is replicated and amplified during infection. At the same time, this polymerase synthesizes subgenomic RNAs. Again, this is in the case of MHV. There are 6 of them, and these are minus strand. These are negative strand RNAs, and they're synthesized by a really complicated process whereby the polymerase will stop at a transcription regulation sequence and then jump to the 5' end of the genome so that it synthesizes an RNA, shown here, with copying the 3' end of the genome and then adding onto it this so-called leader sequence from the 5' end of the genome. And in some cases, it'll transcribe up till this TRS and then make this RNA, et cetera, so that a set of subgenomic minus strand RNAs is synthesized all with a leader sequence or anti-leader sequence on their 3' end. These are then copied back. These are subgenomic negative sense RNAs. They're copied back into positive sense messenger RNAs, all with the same leader sequence. So this complicated process results in 7 mRNAs, including the genome. SARS has a few more than that, actually. So these are the RNAs, the blue ones, that actually are translated into protein. Okay, so I just want to talk just quickly about RT-qPCR detection of coronavirus RNA, because I think everybody knows by now that that's the diagnostic test for coronavirus RNAs, detections of genome from clinical samples. And so this is something that's important to think about. So so here are the RNAs again. This is a gel from my lab from 1983 that I found from a paper we published way back then. And this is a gel showing those messenger RNAs. Here's the nucleocapsid. This is the most abundant RNA shown here. It encodes the nucleocapsid gene. And this is the genome RNA. And then these are all the intermediate subgenomic RNA shown here. So when doing RT-PCR, many of these tests use primers that represent the replicase or the RDRP. So that would be shown, that would be amplifying sequences right in here. And that would amplify only the genome RNA. And then there are also tests using the nucleocapsid gene primers, and those would capture all the mRNAs. So while PCR for RDRP would give you genome copies, PCR for the nucleocapsid would give you copies of both genome and all the RNAs. So this would be good for quantifying from actually infected cell RNA, where RDRP would be detecting free genome RNA. So it's just important to think about what you're testing for when you use these primers. This would be a more sensitive test to look for the nucleocapsid, because all of these RNAs have nucleocapsid sequences, and you can see it's also most abundant RNA. Okay, so now let's talk a little bit about coronavirus proteins. So here again is the genome. we have all these RNAs that I talked about before, and each one is translated from its 5' end into one, usually one protein. Occasionally 2 proteins are synthesized from overlapping open reading frames here, shown here. But the 5' end, or the 5' 2/3 of the genome, is translated into these 16 non-structural replicase proteins, and it's translated through polyprotein 1A and polyprotein 1A, 1B. And what regulates the ratio of these 2 proteins is a translational frameshift sequence, which is located at the end of PP1A. And this is really interesting. What happens is that there's a structure in the RNA called a pseudoknot. And what that does is it causes the ribosome to kind of stall, so that while most of the time the ribosome translates all through here and terminates at this stop codon here, synthesizing PP1A, at some frequency, maybe like even 20% of the time, the ribosome stalls here and at this sort of so-called slippery sequence and moves back 1 frame, so that translation continues through this termination codon and synthesizes PP1A, 1B. And this ratio of these 2, PP1A and PP1A, 1B, is important. If this is altered in some way, the virus becomes highly attenuated. So I always think of this as a possible site for intervention and for attenuation of virus for some kind of antiviral therapy. But I want to talk about these coronavirus, these replicase proteins in more detail. So as I said, the replicase locus encodes 16 conserved non-structural proteins that's shown here, NSP1 through 16. The 3C-like protease is encoded here, and that's the main protease that processes most of this polyprotein. As these proteins are being synthesized, they're being processed by this 3C-like protease and also by a papain-like protease shown here, which processes these 2 cleavage sites. And then, of course, the RDRP is found in NSP12. This is the RNA-dependent RNA polymerase. And these proteins have been certainly talked about as targets for antiviral therapies. Remdesivir targets the RDRP, and lopinavir, ritonavir, these are 2 HIV drugs. This is actually an Ebola drug repurposed, and these are HIV drugs that also are targeting 3C-like proteases. I'm sure there are clinical trials being going on with these drugs now, and we don't know how successful they're going to be. So just again, there are proteases that process replicase proteins, and RDRP that synthesizes genome and RNA, and all of these are potential targets for antivirals. But there are a lot more targets. Coded also are a primase and a helicase. These are both sort of needed for aiding the RDRP in replicating the RNA. There's also the proofreading enzyme I mentioned before, XON, and a guanylyl transferase that's important for mRNA capping. There's ENDO-U is another endonuclease that's an important host antagonist of host innate immune response, and another transferase here shown, another methyl transferase important for capping. So these are all extra activities that are there to promote synthesis of viral RNAs and capping of the 5' ends, which is important for translation, and also for protection from host cell sensors and interferon responses. And so all of these, again, are potential targets for antivirals. And then finally, there's NSP1 that promotes host mRNA degradation, translation of host mRNA inhibition, cell cycle arrest, inhibition of interferon signaling, many activities to protect the virus from the host response, and even more. This is an ADRP or macro domain, and a deubiquitinating activity. Again, additional activities to antagonize or evade host cell responses. And just to show one example how this kind of approach would be really good as antivirals against multiple viruses. Here's an example of NSP13 or helicase inhibitor. So these are molecular models of the helicases of SARS, MERS, and MHV. And this compound, SSYA10001, fits into the same groove of each of these structures, binding pocket, and then also inhibits replication of each of these viruses. And as you can see here, we're plotting viral replication on the y-axis as a function of drug concentration. And in each case, the virus replication is inhibited by increasing concentrations of the drug. So here's just one compound that can combine or can inhibit the helicase of each one of these coronaviruses. So this is, I think, a really important approach going forward if we're going to be prepared for future coronavirus pandemics or epidemics, or even any kind of viruses. So this would theoretically work probably against all coronaviruses. So in summary of that section, there are many conserved features of coronaviruses. The subgenomic mRNAs, the non-continuous RNA synthesis, the ORF1A, 1B frame shifting, these conserved proteases, the RDRP and RNA modifying enzymes, and all the host antagonist activities. Just for example, some of these are here. So I think most of the activity's been targeting proteases and RDRP, but all of these other proteins are really possible targets as well. Okay, so the third part of the talk is going to be a little story from my own lab, but just to preface it, I want to talk a little bit about the host innate response. So during infection, all RNA viruses really, and some DNA viruses, produce double-stranded RNA, which of course for these viruses is part of their replication cycle in replicating their RNA and transcribing their mRNAs. So this viral double-stranded RNA is recognized by the host cell by sensors, including MDA5, which you've probably heard of, which signals through MAVs to produce interferon type 1 and type 3, to produce interferon stimulated genes, products which are antiviral. So this is a pretty canonical antiviral pathway. But there's another pathway in which double-strand RNA is perceived or sensed by oligoadenylate synthetases, which then synthesize 2,5-A, a small oligoadenylate with an unusual linkage, 2', 5', that triggers dimerization and activation of ribonuclease L, which then cleaves cellular and viral RNA, which leads to protein synthesis inhibition, increase in apoptosis. And a third pathway, PKR, protein kinase R, PKR also senses double-stranded RNA, leading to phosphorylation of PKR, and then phosphorylation of IF2 alpha, a protein synthesis initiation factor, which shuts down protein synthesis. So many viruses, or I would say just about all viruses, attempt to shut down these pathways. And I'm going to talk about one story from our lab in which, I should say also that the interferon pathway activates or upregulates these other two pathways as well. It's kind of the central pathway. But I'm going to talk about one story that shows that how coronaviruses have some coronaviruses completely shut down this pathway. Because we know that that even if interferon pathway is shut down by many viruses, this pathway can still be activated, and so can this one. So some viruses have designed proteins to specifically knock down this pathway, and that's what I'm going to talk about. So the enzymes that shut down that pathway are called 2', 5' phosphodiesterases. They cleave 2,5-A, the activator of RNA cell, and they're encoded in lineage A and lineage C coronaviruses, not in SARS-CoV-2. And they're encoded in this NS2 protein or NS4 protein, NS4B, or in toroviruses, related nidoviruses in the polypeptide encoded in ORF1A, PP1A. So this is sort of interesting. So this is the only case, I think, where there are homologous proteins of accessory proteins in two different lineages, and they're actually located in completely different places in the genome. So it's kind of interesting to wonder where they came from, because they probably were derived from host cells at some point. So these proteins are 2', 5' phosphodiesterases. They're members of a 2H phosphoesterase family shown here, which includes cellular enzymes shown here. And all of these proteins have similar HXTSX motifs, two of them, about 90 amino acids apart. Otherwise, they're not that homologous. And other than coronavirus, this is another sort of interesting fact, that only rotaviruses encode such a protein as this. So rotaviruses, double-stranded RNA viruses, are completely unrelated to coronaviruses, yet they share a homologous protein. This is just to show, these are just these are models of the tertiary structure, the crystal structure of these phosphodiesterases. These are actual structures for ACAP7, and this is the cellular protein, and MHV-NS2, and this is the MERS protein. And if they're all, this is just a predicted structure. If they're laid on top of each other, you can see that they really very, very similar proteins. Okay, so let me talk a little bit about the function. So NS2 is an accessory protein. This is the MHV lineage A genome. OC43, the human virus, also encodes this protein. And here it is with its 2 catalytic histidines. So we very early on were able to get a wild type and mutant virus of strain A59. So where I say A59 in the next few slides, it's actually the wild type virus. The mutant is NS2 with this histidine inactivated so that this enzyme is no longer active. So we looked at replication of this protein in bone marrow derived macrophages. And although NS2 is not required for replication in mouse fibroblasts, it's very much required for replication in macrophages. And these are cells that have an active innate immune response. The mutant virus is really unable to replicate, while the wild type virus replicates to quite high titer. This is a log scale of replication. So this is a difference of maybe 10,000 fold difference in replication. However, if we replicate the viruses in macrophages derived from RNA cell knockout cells, replication is recovered, suggesting that the inability to antagonize RNA cell is what causes the attenuation of the mutant. And if we look at ribosomal RNA degradation, this is the way we assess RNA cell activation. In the presence of activated RNA cell, we can see with these mutant infected cells that the ribosomal RNA is degraded. This is the normally 28 and 18S RNA that you can see intact in the wild type infected cells, but degraded in the mutant infected cells, suggesting that RNA cell is indeed activated in here where the virus is unable to replicate. We did the same kind of experiment in a mouse. We infect mice intrahepatically. Virus replicates, peaking at about day 5, and then is cleared by the CD8 T cell primarily. But during the early infection, the innate response is quite crucial. And when we looked at replication at virus in the livers of these mice at day 5, the peak of infection, we can see that virus replicates quite robustly to about 7 logs. That's 10 million PFUs per microgram of tissue, whereas the mutant virus barely replicates. But if we do the same infections in RNA cell knockout mice, the mutant virus can recover replication, consistent with what we saw in cells. And the same thing was true. We could substitute NS4B of MERS or OC43 of NS2 OC43, and that would have the same function of NS2 in the mouse. So, okay, so this is these are tissue slices stained for viral antigen from these same mice. And we can see again that there's quite a lot of replication in B6 and knockout mice by wild type virus, but the mutant virus only expresses its antigen in the RNA cell knockout mice. And the same is true for inflammation and by staining with H&E, we can see that again, that we see really no pathology at all in the mutant in the wild type virus, wild type mice, whereas the mutant in the knockout mice can cause inflammation and necrosis, similar to the wild type virus. So just to summarize, the double-stranded RNA is perceived or sensed by OAS, which produces 2,5A. The phosphodiesterases of these coronaviruses reduces the 2,5A, degrades it. So, and that's true for coronaviruses, MHV, OC43 MERS, toroviruses, rotaviruses. RNA cell is not activated, and virus replication and pathogenesis proceed. So in the case of this mouse model, this one small accessory protein is actually absolutely required for replication and pathogenesis. And just to point out now the accessory proteins of SARS and SARS-CoV-2, this is SARS. It has 3A, 3B, 7A, 7B, 8A, 8B, 9B, and maybe 10. So quite a few of these proteins. ORF3A is a channel protein. ORF3B seems to be missing in SARS-CoV-2 relative to SARS. ORF6 is a really interesting protein that interferes with nuclear translocation of STAT1, and that would really cut down on interferon-stimulated gene expression. The proteins of SARS-CoV and SARS-CoV-2 are somewhat different in ORF6, so that's something we've been interested in. ORF8A is kind of interesting. It's ORF8A and B in SARS and SARS-CoV-2. There's only an ORF8. Originally, SARS-1 had only an ORF8, which then kind of adapted and accumulated a deletion, which led to an ORF8A, ORF8B proteins. So we're watching this region to see if it will also change in SARS-CoV-2. And I'm not sure that this ORF10 is actually expressed, but these proteins are quite interesting, and I'm sure there's going to be a lot of studies on them. Okay, so coronaviruses are adept at antagonizing innate immune responses. Each lineage encodes a unique accessory protein set of accessory proteins that antagonize the antiviral response, and they're often redundant in function. I didn't really show that, but sometimes we can knock out one of these proteins and there's another protein that will have the same function. So lineage B beta coronaviruses encode 2'5' phosphodiesterases that antagonize the RNA cell pathway. And for MHV is an organ-specific virulence factor. I didn't show this, but NS2 has no function in the brain, the other target of MHV, but only in the liver. And the SARS coronavirus 2 accessory protein shows some differences from the original SARS. And we will find out whether these are significant for pathogenesis. Just really quickly, some of the models that have been used for coronavirus pathogenesis, initially, we would start working with human lung-derived cell lines, then progress to primary lung cells, which we're starting to do with some of our colleagues at Penn. Then the people are making transgenic mice, transgenic for human ACE2 protein. Mice are not inherently susceptible to SARS-CoV-2 infection. There's been a lot of work on a hamster model, which seems to work pretty well. Also ferrets and non-human primates. So there are many models to study this virus. This work is just beginning. There's also, we've been interested in studying the virus in the bat as well. And with a collaborator at Colorado State, Tony Schounts, he has a bat colony. And we've been doing experimental infections of MERS and SARS-CoV-2. And just one piece of data that Tony found that after 3 weeks of infection, this is in ELISA where he measured the antibody titers against nucleocapsid protein. And there's quite a good response against nucleocapsid in this bat here, in this bat as well. And the reason that bats are really interesting is that they harbor many pathogenic viruses, but don't seem to get sick from them. So it's a really important or interesting question of what's different about the host response in bats. So these bats didn't get sick at all, yet they did respond quite well with an immune response to SARS-CoV-2. So this is, again, just the beginning of a study. We hope that it'll tell us something interesting about the virus. And I'm just gonna acknowledge my lab. Shown here, and these 4 people in bold have been working very hard now on SARS-CoV-2 in our BSL-3 facility while I stay at home and work at home. I just acknowledge our long-term collaborator, Bob Silverman, who we do all our RNA cell work with, and Ralph Barrick, who's we've made some mutants of MERS. So I will stop there.
Thank you very much for this inspiring talk, Susan. Thank you.
Thank you, Matthias. Thank you, Professor Weiss, for that very interesting presentation. So the Q&A session is now open. We have a few questions in the Q&A sector. So Jan Ming has been very active and has asked quite a few questions. The first one is, do coronaviruses survive the stomach and intestine environment to cause GI diseases?
Ooh, well, some coronaviruses are GI viruses. This virus, I hear people do get GI infections and diarrhea from this virus. How they survive is a good question because, yeah, I I don't really know specifically, but it seems like they must be surviving to some extent. Sorry, I don't have a more definitive answer.
No problem. Then her next question was looking at the genome of the lineage A, B, C. Anything special that may explain the differential pathogenicity in humans?
You mean between the 3 different lineages? Is that what she's saying?
Yeah, yeah.
You know, not really, because you can see from the stuff I talked about at the end that like one little protein and one amino acid can make a huge difference in the pathogenicity. So it does look like the lineage B viruses, the SARS-like viruses, have a more complex array of accessory proteins. But the MERS virus is in lineage C is really just as pathogenic as the B viruses. So I would say no, you really can't tell. And the vast majority of the genome, the replicase 16 non-structural proteins are quite conserved among all the viruses. So I think it's not at all obvious from the genomes.
Okay. Then the next one is, do you know bat coronaviruses have the furin cleave site?
Okay, so that's the bat coronaviruses that are related to SARS, SARS-1 that is, the original SARS, do not have the furin cleavage site. They look a lot like SARS. The bat corona, this RATG13 that I talked about, that's clearly the closest related virus that we know of to SARS-CoV-2, does not have the furin site. Whether there are any bat viruses with furin sites, I don't really know that. I'm guessing there are because MERS has the furin site, and there are definitely, there are bat-related, MERS-related bat viruses that most likely do have the cleavage site.
Then we now have Evelyn Bischoff, who has raised her hand and has a question. I will let her speak. Evelyn, hello. You can speak.
It's such a great honor and pleasure. Let me just say that you are my legend for sure.
Oh my God.
I was wondering, based on a fantastic speech and so many questions, but I will just ask one. Do you think, or do you have a hope that based maybe on the animal transmission experiments, there will be a way to say or to predict if seropositive human individuals are actually transmitting the disease, and at which extent? That's
a tough one. I mean, I would think that a better way to do, not so much seropositive, but positive by genome positive or virus positive. But it's tricky because I know from our experience, like looking at clinical samples from our hospital, we find that there's not a whole lot of genomes in the patients late in infection. So I would imagine that somebody sort of towards the tail end, when they have developed antibodies, probably don't have much virus available. So I don't think somebody with neutralizing antibodies is likely to be shedding virus and spreading it. But I don't know how to tell for sure.
Thank you so much.
Yeah, you're welcome.
The next question is from Xupeng Hong, who asked why the pan-RDRP inhibitor ribavirin does not used for coronavirus treatment.
Because I think it doesn't work. I think it's just that ribavirin's a nucleoside analog, correct? I think it just may not work with that RDRP.
Okay, thank you.
I'm not sure the mechanism of ribavirin. Maybe I'm wrong about that. But whatever it is, it doesn't work on this particular virus.
Okay. Xupeng has asked another question, which is how frequent the recombination happens for coronavirus. Some studies showed furin cleavage is not essential for coronavirus cell membrane fusion, but enhances cell-to-cell fusion. Does that contribute to different pathogenicity of SARS-CoV-2 and SARS-CoV?
Okay, there's kind of two questions, and I'd like those questions. So recombination happens a lot, okay, and so there's certainly a speculation that SARS-CoV-2 is a recombinant virus, but nobody's found like the telltale parents that it might have come from. So we don't really know about that. And as far as the furin site, I think the furin site's really interesting because, for example, the mouse viruses, some there are a lot of different mouse virus strains. Some have a furin site, some don't. They're all completely pathogenic. So I don't think that whether it has a furin site or not tells you that it's pathogenic. It's not required because there are other proteases on the cell surface like TIMPers or even and other ones as well. For the mouse virus, it's a zinc metalloprotease that can mediate that cleavage for viruses that don't have a furin site. So it's really not required, but it would kind of push the virus towards the endosomal pathway of entry rather than the cell surface pathway. But yeah, so in my mind, it's not at all a marker of, it's not like because SARS-2 has the furin site, it's more pathogenic than SARS-1 that doesn't have the furin site, or that it spreads to different, I mean, it could affect spread to different cell types, but we don't really know that.
Thank you. Maria Joao Amorim has asked, the immunity of humans to circulating coronaviruses is not long lasting. What is your view regarding SARS-CoV-2 immunity?
Okay, so I'm not an expert on clinical immunity, but I guess I've heard the same thing, that OC43 people can be infected again and again. I think that there definitely is an immune response because all these recovered people have been shown to have responses. And in fact, they're using plasma transfer for a therapy for patients. So there is an immune response, whether it peters out a little bit or somewhat, and we don't know the kinetics of that. But I still think that if someone were to be reinfected, it's be kind of like after a vaccine, that you might get not nearly as sick if you're reinfected. So I would imagine that SARS-CoV-2 is going to be, there will be immunity, and maybe it'll wane over time. I don't really know, but I don't expect it to be that different from some other viruses.
Okay. Zilong Yang has asked, which non-BSL-3 human coronavirus is a good model for basic viral replication study?
Oh, I think MHV is. I've worked on it for like 30 years. Well, MHV, people worked on MHV a lot, on IBV. There's a lot of work on the chicken virus, BCV, bovine coronavirus. The human viruses are also the OC43 and 229E. They're all BSL-2. They're a little bit fussier to work with. The mouse, to me, the nice thing about the mouse virus, on top of it being really easy to work with, is that it's got a great model in its natural host. Like, you don't have to force it into a mouse. It's normal, it's natural to go into a mouse. And the other human coronaviruses, really no mouse, there's no models for them really, 229E or OC43.
Okay. And maybe the last question, and then all the questions that have been not yet answered, Professor Weiss has kindly accepted to give you a written answer to them after the webinar.
Don't make them too long. Don't make them too long.
It is, what do you think the main viral evasion pathways are of CoV-2? You mentioned a few, and do you think this is what makes it so pathogenic and easy spreading?
Well, I don't think, I mean, it's actually, if you talk about virulence and lethality, it's actually much less lethal than SARS or MERS. So, but it spreads much more readily. So nobody really understands anything. I mean, we understand that it spreads because people are walking around asymptomatically shedding virus. So it's like different, it's almost like a different phenotype from SARS, from the original SARS, but we don't really know why. There's nothing obvious about its replication or anything like that that would tell us that. So maybe it does have to do something with some of the host antagonists, but that's going to be a really hard thing to prove. I mean, all the coronaviruses shut down interferon pathways pretty effectively. Whether this one, we don't really know enough about this one yet. That's one of the things that my lab is interested in understanding, like on molecularly, what does it do to the host innate response? So I didn't really answer that question because I don't know the answer, and I don't think anybody really knows the answer, but maybe we'll learn more.
Okay, great. Well, that closes our Q&A session. Thank you very much.
Thank you.
Thank you very much, Susan. It was a real great afternoon, and
morning.
We will yes, morning, sorry, or evening in China. So
yeah,
and so we've had a lot of people watching, and it was also live streamed on YouTube, and so we will send you open questions, and we stay in touch.
Okay, thank you.
So finally, I would like to say that from the 11th of May, Switzerland is open again. So that was just decided during our talk. So restaurants will go open, museums, kids have to go back to school, etc. And they will now test whether there is a second wave or not. And we'll give 2 weeks until the 27th. And so we will go back to normal now.
Wow, not here.
That was okay, but soon, I hope.
I don't know. I'm scared.
Yeah. Okay. So thank you very much again, and everybody, a nice evening, morning, or afternoon. Bye bye.
Thank you.
Thank you.
Bye.
Bye bye. Bye bye.
Bye.